So today we're going to talk about close reading strategies in science, and we're going to be using some of the material over Unit 2. And so we're going to talk about close reading of textbooks and also close reading of science articles. So just some general tips to remember for close reading in the sciences. First of all, it often involves reading at the sentence level and often with multiple passes, so that they'll read at one time with one purpose and then maybe reading it at another time with another purpose. There's a big focus on integrating ideas. So in science, that means looking at the text and then the diagram to text, and also reading across related topics. So a lot of times when you're close reading in science, you need to go back to a previous section or diagram to make sense of what you're reading right now. And I'll show you a couple examples of that as we go. And the goal for most of the reading in science is to understand that science process being discussed and how to relate it to previous science processes learned. So as they're reading, making those connections is going to be pretty important. So the first example, this is how it looks in the text. This is from Phelan Chapter 5, which is the textbook example from Unit 2. So I know you can't really see this here, but you can see that at the bottom there's 5.5, that section. We're going to start with that and then move to the diagram and the text on that second page. We'll come back and refer to the diagram on the top of the first page as we read the second page. So students will start out by reading the title, which is How Do Genes Work? An Overview. And something that they need to think about here is that word overview. So it's giving them the general ideas of how genes work, and then as we get deeper into the chapter, it's going to really explain the two processes of transcription and translation that are provided in this overview. It's also important for students to be looking at those two headings at the bottom there, biotechnology and agriculture and DNA sequences. And remember, throughout this particular chapter, those bars at the bottom there tell the students sort of the categories, where this fits into the larger picture of DNA processes. So even the textbook chapter is trying to help them fit that information together, and it's something that they want to pay attention to. So to start out a close reading of this particular section, read the first sentence aloud to students, which is, just as having a recipe for chocolate chip cookies is not the same thing as having actual cookies, having a wealth of hereditary information on how to build muscle cells or root cells or leaf cells is not sufficient to produce an organism. And then stop and talk about what that means to students. So what are they getting from, that analogy of chocolate chip cookie to chocolate chip cookie recipe in terms of DNA material and having actual cells? So think about it. Every cell in an organism contains all of the information needed to manufacture every protein in its body. This means that the skin cells on your arm contain the genes for producing proteins found only in liver cells or red blood cells or muscle tissue, but they don't produce them. Having the instructions is not the same as having the products. That's another good time to stop and ask students about what they think this means. So we're using analogies here of recipes and cooking, but students can extend that a little bit to think about, what else do they know? Where you have a plan, but you don't really have the whole thing. And they might bring up, instructions for building furniture or something like that, where you need to have more than just the plan to have the actual product. So the genes in the strands of DNA are a storehouse of information, an instruction book, but they're only one part of the process by which an organism is built. And let's move on to students can start to pull this together. So this is an overview of how genes work, and students need to do a close read of this figure as well. So it's starting out talking about, the genes themselves and DNA as a grandmother's cookbook, right? So these are the instructions, but it doesn't really tell you what's happening. And so then students need to look at that second sort of panel where it's showing that copying the recipe is sort of like transcription. Whereas at the top 2 down where it says translation, that's different than just copying it. That's combining and baking the ingredients and putting it all together. So this is sort of giving them an overview of the whole process of taking a strand of DNA and making it into a new protein molecule. So we're going from text to the figure, and now we'll move back to the text. So looking at a particular trait, its genotype, is like a recipe for a cookbook, the physical manifestation of the instructions. Whereas an organism's phenotype is the cookie or the macaroni and cheese or the French toast or any of the other foods described by the recipes. So now let's go back and look at that. Where is it talking about genotype? So that's to ask students to look at that part, and that's where it's talking about that cookbook. And then look at the phenotype, and that is any of the foods that are described by the recipe, right? So that is the actual manifestations of whatever instructions it was. So that's at the very end there. That's that protein molecule that you end up with. And along the way is the process that you have to get to those final protein molecules. Back to the text, ask students to read the rest of that section, thinking about going back and forth between the diagram and the text to look at, that next paragraph. How does a gene, a sequence of bases in a section of DNA affect a flower's color or the shape of a nose or the texture of a dog's fur, which is that phenotype. And so it occurs in 2 main processes: transcription, which is a copy of the gene's base sequence, and translation, which is that copy used to direct the production of a protein. So have them go back to that diagram and look at where that's occurring in there. And then right here at the end is the call out of that figure. So that figure that we've been looking at, this is where there's a caption for it, which says that it presents an overview of the processes of transcription and translation, and then defines each one. So students need to understand the overview of this process. What are these 2 things, transcription and translation, getting us when we look at DNA? So understanding those processes before the textbook goes deep into it is one way they can do a close read. Then they'll move on to the processes of transcription and translation, which the textbook does a really good job of breaking down. They may want to go back to this diagram to make sure they understand that whole overview. In addition, where it says on the figure that in transcription, the eukaryotes occur in the nucleus, the gene's base sequence or code is copied to a middleman molecule called a messenger RNA. That might be a really good time to return to an early. This is the diagram from earlier in the chapter. It's figure 510. We were just looking at figure 511 that talks about coding and non-coding DNA, but also shows the eukaryotic cell and the nucleus and exactly where these DNA sequences, base sequences, are housed. So it gives students a good sort of another in-depth picture of what's going on. Then they can return back to looking at this caption of the figure to think about, well, what does it mean in terms of that metaphor that's been running through of a recipe, of a cookbook? And how does it make sense now that they have seen a picture and they've read it in depth? They'll be ready to read more about DNA and, transcription and translation of DNA. So you would continue doing a close reading of those sections as students go through. And again, they may be just reading it to talk it out loud like we were in that section, or they may be reading it to annotate as they will be in later sections. I wanted to give you a second example, and this is also from Unit 2, and this is on reading a research article for argument. So there's many different ways that they've been presented reading a research article. Sometimes it's just to read the abstract to get an idea of how the research was done. In this particular case, they're reading for what is the argument being presented. And so it's a good time to remind them about the parts of an argument, which is a part of this particular lesson in Unit 2. So they'll be looking for the data, which are the facts involved in the argument that support the claims, the claim, which is the conclusion that's drawn from the data, the warrants, which are the reasons that justify the connection between the data and the claim. So in science, these are often the scientific principles or the methods, and the backing. And these are the basic assumptions that are commonly agreed upon that justifies the warrants. So their ultimate goal for reading this particular article that we're going to look at in a second is to present the argument as a sentence. So the author argues X because, and that's where the data is, since, and that's where the warrant is, on account of this particular backing. And although some believe or are concerned about some counter-argument, the data suggests X, therefore the conclusion. So they're going to be reading the whole article to be able to summarize it in this one sentence argument support. So they're going to be reading an article out of Nature Education. And so the first thing that we want them to do is read the title, which is Genetically Modified Organisms, GMOs, Transgenic Crops, and Recombinant DNA Technology. So ask them what they know about GMOs and how they've heard about it before, and then have them read that sub-caption, that subheading. If you could save lives by producing vaccines in transgenic bananas, would you? In the debate over large-scale commercialization and use of GMOs, where should we draw the line? So let's think about that in terms of the title and in terms of that subtitle. What might the author be arguing? And so students can start doing a close read right there at the very beginning. The way that this is presented, it says saving lives. So what might the argument be? So students can start to use that information as they read the beginning. So for this one, ask students to read the first paragraph of the article and then explain what is the author discussing. So the first paragraph says, people have been altering the genomes of plants and animals for many years using traditional breeding techniques. Artificial selection for specific desired traits resulted in a variety of organisms ranging from sweet corn to hairless cats. But this artificial selection, in which organisms exhibit specific traits that are chosen to breed subsequent generations, has been limited to naturally occurring variations. In recent decades, however, advances in the field of genetic engineering have allowed for precise control over the genetic changes introduced into organisms. Today, we can incorporate new genes from one species into completely unrelated species through genetic engineering, optimizing agricultural performance, or facilitating the production of valuable pharmaceutical substances. Crop plants, farm animals, and soil bacteria are some of the more prominent examples of organisms that have been subject to genetic engineering. So to do a close read of this, students could look at what are they talking about in terms of artificial selection. What do they know about that? What does that mean? And what does it mean to naturally occurring variations to genetic engineered variations? And talking about what that might mean and what some of the problems might be with that kind of technology. So that can be a whole class discussion of a read. You can add that silently or they can read it aloud. And then move on to the next section where we look at the current use of genetically modified organisms. So this is where they're going to be talking about GMOs. So we can start out with that first sentence. Agricultural plants are one of the most frequently cited examples of genetically modified organisms, GMOs. And we can look at that figure 1 to be thinking about, what does that mean? What are we talking about here? And even before we get to that call out of that figure, students will be able to see that we have a little tiny fish at the bottom and a much, much bigger fish at the top. So perhaps that's what they're talking about there. It goes on to talk about some benefits of genetic engineering, increased crop yields and so forth. So for this particular section, ask them to read the section on the current use of GMOs to themselves and mark up the argument. So what is being argued here? And in general, they'll be talking about the benefits, right? So the benefits of genetically modified organisms. So as they work through that together, make sure that they look at the table. So show some examples of current uses, already approved uses of GMOs. So we have herbicide tolerance for soybeans, we have insect resistance to corn, and it gives you the genetic change that's in there. But most important for students to understand at this point is that there are some things that are already approved that are being done right now. But if you also notice that salmon example isn't here. It's not one of the ones that are already approved. Back to that particular paragraph where it's talking about the salmon. So at the bottom sentence it says, for example, salmon have been engineered to grow larger, that's figure 1, and mature faster, which is table 1. And cattle have been enhanced to exhibit resistance to mad cow disease. So let's go back to table 1 because that first section didn't show the example of salmon till we can. So this is under the section in table 1, products still in development. And so it's faster maturation. And if you look at the coho salmon example, it says a type 1 growth hormone gene is injected into fertilized fish eggs. So this has yielded faster rates, but it's not yet been approved for use. So this is one that's still in development. So that can tell students a lot about this field. This is a burgeoning field. New things are happening. It's something that we're going to be looking at and they'll be looking at as their final project. What are some newer things that are happening with agriculture and DNA? So close reading for the argument, they'll be looking at the paragraphs, thinking about that warrant, data, backing, conclusions, to be thinking about how can they pull out the whole argument from the whole article. And the article goes on to discuss not only what are the benefits of GMOs, but what are some of the concerns and what are some of the drawbacks. And overall, it's a fairly positive view of GMOs. They'll be reading some other stuff that may take a more negative view of it. But what they need to do is pull out what that author is saying is the argument. So it's not their own argument, what they think is true. It's what is the author presenting as true. And that's in science what they need to do to pull out that argument. It's not their interpretation of the argument. It's what is being supported in that particular article. So there's a bunch of different strategies for close reading in science. So a few examples might be text annotation, might be paired reading, might be reading aloud as a whole class or taking turns reading. It might be reading silently for discussion. It might be reading silently to prepare an argument or any combination within. And it depends upon the purpose. For some things, students are reading to prepare for their final project. For some things, students are reading to prepare for an exam. For some things, they're reading just to talk about it with each other to make sure that they understand the points that are being made. So the strategies that they'll be shown and then the strategies that they eventually will self-select depends upon what they're being asked to do. And that is a brief example of close reading in the sciences.